Hello and welcome to Talking Points. I'm Dave Kelly, Director of Advanced Media Production at Cal State Long Beach. Our topic today is genetic research, genetic manipulation, and the bioethics related to those areas. My guest today is Dr. Lisa Klig. Dr. Klig is a professor of biological sciences, and she is also the director of the Biotechnology Certificate Program. Welcome, Lisa, and thank you for joining us on Talking Points.
Thank you for having me.
Well, before we talk about manipulation of genes and so forth, we need to first talk about some very basic science so that we can put this in context. Most people know about DNA. They hear about it in terms of its use in criminal cases to try to use the matching tool of connecting the culprit to the crime scene. They also have heard about DNA testing in terms of creating a match and to, I guess, process identity to see if we really are related to people that are quite famous. But other than that, DNA and the genes play a very significant role in disease. And what is that role that genes play in disease?
Well, okay, genes are known for being possibly changed. The word we usually use is mutation. But let me first explain to you what a gene is. Most people know we have chromosomes. There are 23 things in our cells. And each of these chromosomes are made up of A's, T's, G's, and C's, the letters. And so these letters are arranged in a pattern, like a book. So if you say that each of our 23 chromosomes is like a sentence, then we all know sentences are made up of words, so that each one of these words is comparable to one of our genes. Now, these genes are made up of the letters, the A's, T's, G's, and C's that we've all heard of. But what happens if a letter is changed? Well, if you're looking at a book and you see the word flat, F-L-A-T, change one letter, you got fat. Might not be a good thing, or it might be a good thing depending on the sentence. So again, 23 sentences made up of these words. Each word is the same in my world as a gene. It's made up of letters. So when you get these changes, they could be good, they can make you much better or stronger, or they could be bad and cause cancer or other diseases.
So we're talking about mutation now. So yes, mutation is something that occurs during the replication process, so that once a mutation occurs, replication from that point on will reflect that mutation.
Yes, once there has been a change made, that change stays. So if it's made early on in your development, it'll be all over your whole body. And if it's something that makes you bigger, stronger, better, that's great, you're the lucky one. But if the change is something that is deleterious, that causes a disease, then it's not so fortunate.
But now we have new technologies and new techniques to identify the genetic code, to look at the various sequences, and we can actually do something about those sequences in some cases. Tell us about that.
Okay, so now you're first talking about just getting the order of all the letters in all of my 23 sentences. That's what your sequencing is. So now we have all the letters of our book, but you have to remember that's one cell in your whole body. So the first question you got to ask is, so how much of this book is actually read to make you what you are? And by that, what we're talking about is turning on genes. Best guess, we're looking at, well, less than maybe 5%. So what we're saying is we got a whole ton of words, we don't know what they do. We got something like less than 5% that are even words we can recognize and read. So those are the genes that we know about. Now we get into the real tricky bit. The ones that are on in the different parts of our body are all different. I mean, everybody knows your skin doesn't look like your liver doesn't look like your lungs. What's the difference? I can tell you that in all of these cells, it's the exact same 3 billion A's, T's, G's, and C's in the same order. And the difference is which genes are on. So we're stuck with the question, what does it mean to be on? And the only image I can give you, easy image, I think. You have an art gallery. You have 2 rooms. Both of them are pitch black. In these 2 rooms, you have 10 identical lithographs. So 10 here, 10 in your other room. Room number 1, you're going to turn on lights 1, 3, 5, 7, 9. Room number 2, you're going to turn on lights 2, 4, 6, 8, and 10. Walk on in. Those 2 rooms look completely different, yet we know they contain the exact same artwork. That's what's going on with your genes. So that your liver may have some genes on, your lungs some other genes on. So now when you're asking me questions about mutations, well, it depends which gene is changed, whether it's a gene that happens to need to be on in that particular place in your body.
So we can identify, in some cases, these genes that are on or off, and if so, what can we do about that?
We can identify the genes on or off, and some of what we could do if we wanted to, and this gets into the more, how shall we say, newer technology. We can either change the gene, we can remove the gene, we can add a second copy of the gene, or maybe we can just change the amount of on or the amount of off, like a dimmer switch, if you want to go there. So now we're moving kind of into stuff that's been around about 2-3 decades, but coming closer to where we are here. Gene therapy came out in the 1990s and then became really popularized around 2008-ish, when it really was one of the first cures for blindness. And that's just a totally amazing idea that we can cure someone who is blind by replacing a gene. But is it really a replacement? Well, not exactly. It's adding an extra gene. So here I'm now stuck with two thoughts. Thought one, are we curing all blindness? No. Blindness is caused by lots of different things. It has to be caused by this very one specific gene that we can then fix this one gene. So that's how the beginnings of successes with gene therapy started. The next issue then is, well, okay, that was just adding an extra gene, but you still have the other one there. And you end up with a question of, for lack of a better way to put it, it's like suitcases. How did I add my extra gene? If I'm carrying the gene in a really big suitcase, I can carry the whole gene. It's really clunky and cumbersome and I may not get it in. Try running through an airport with a big suitcase. If I put it in a teeny little backpack or a carry-on bag, well, I'm not going to be able to fit the whole gene, but I'll be able to move it in better. So I was having problems, people were having problems with how do you move these genes in and out. And a new technique has been really moving ahead since about 2013. It's been around longer, but 2013 was the big breakthrough. And that's called CRISPR-Cas.
And that's referred to as clustered, regularly interspaced, short palindromic repeats. That's the acronym for that long phrase.
Yes, and I don't think you want me to go further than that on that. But what you can do with CRISPR-Cas is you can actually replace a gene, you can actually cut one out, you can stick another one in, you could stick another gene in somewhere else that might modulate the first gene. So in other words, if you have a broken gene that's causing a known disease, in theory, we could actually cut that out and replace it. But that opens all kinds of questions. And there are, of course, some safety issues attached.
So if we can essentially edit the genetic code by using CRISPR, what does that leave us with in terms of ethical issues?
Huge numbers, depending on which way we look. You've just opened a Pandora's box. Because on the one hand, shouldn't we cure the disease? My child is blind and you can fix this. Shouldn't we cure it? Of course. The largest number of trials right now are for cancer. Shouldn't we try to cure cancer? Of course. Yet on the other side, first of all, when you talk about ethical issues, you talk about informed consent, autonomy, you talk about people being able to assess and make decisions. Is this technology ready for prime time, and do we have a way yet to explain it to everyone so that they know what they're buying? And when you go to buy something in a store, you usually know what you're buying. So we need to look at issues about autonomy. We need to look at issues about regulation. How do we know that it's safe, what we're doing?
That was my next question. What about the safety issues? When you refer to safety, what kinds of problems could develop in terms of safety?
Well, with the original gene therapy trials, going way back, this is pre-CRISPR now, the issues had a lot to do with the vectors, the suitcases I was talking about. And a lot of those have been more or less resolved, so that there the issues are, how long does this replacement gene work for? If you integrate it, you glue it in, it'll stay longer, but we may not want to be doing that because that has some issues like where is it getting glued in and what else is it affecting? If you glue a picture on your wall in Los Angeles, it won't fall off in the earthquake. But what if you want to move it later? Meanwhile, the other choice is you can just have it floating there on its own, but when it's not glued in, then it'll over time dissipate, it'll go away, so it won't work anymore. So starting back with those problems, we now move forward to the current problems. What are the issues now? Lots and lots and lots and lots. Does the CRISPR-Cas system work? Yes, definitely. Is it, at the moment, 100% accurate 100% of the time? Certainly not enough experiments have been done to say this safely. I think that we're moving in a direction that would be wonderful to cure diseases, but we have to proceed with caution.
So what I'm hearing you say is that, first of all, we don't know if it's a long-term solution for the reasons you mentioned. Does it have the staying power? And secondly, are there other problems that could develop as a result of this that we're not aware of yet?
Okay, let me clarify. The staying power definitely does. Once you change with CRISPR-Cas, when you cut a gene out, you've cut it out. It's gone. If you stuck a new one in, it's in. It stays. So the changes we're making are permanently in the person. Now, if we're only going into their body cells, their somatic cells, it's in that person but not going to carry on to their children. If we go into the germline, then it would carry on to their children also.
Okay, all right. So this is a new technology. As you mentioned, it's only been out a couple of years now in terms of the work that's been done. It's a work in progress. It leads us to some other issues, which is, let's assume that we develop this, as we will, to a more finite process, or a more well-defined process, I should say. Who's going to decide what is a defective gene and what is not, and what needs to be replaced and what does not?
That was what I was alluding to earlier, that we end up with a terrific number of issues that need to be addressed now. There has to be a lot of public discussion now about who's going to decide, how are we going to decide, who's going to fund these things. I mean, we can't sensibly expect the politicians who are non-scientists to just pass laws arbitrarily. Now, there are some very interesting bioethics commissions that have been formed over the years that are supposed to guide our country. There are also bioethics institutes that are supposed to help us guide it. But still, at the end of the day, I think the public also needs to weigh in, to learn about it, to see what it can cure. I mean, it's amazing, the disease that can be cured, and to see where the possible problems might be.
And it sounds like there are a number of problems. We're going to discuss more of those when we come back from the break. Stay with us. We will come back and discuss more about genetic research and genetic manipulation and the bioethics related to it after the break.
Who designs things like needle-free diabetes care, robotic checkups, and electronic aspirin? The answer: biomedical engineers. It is one of the most rapidly growing career fields in the United States. These people analyze and design solutions to problems in medicine and biology, improving the quality of patient care. With a degree from Cal State Long Beach, you can become a part of this amazing career field.
Welcome back to Talking Points. I'm Dave Kelly. My guest today is Dr. Lisa Klig, and today we're talking about genetic testing, genetic manipulation, and bioethics. And Lisa, before we left, we were talking about what some would refer to as this brave new world of CRISPR. It's still new. Some other thoughts about CRISPR that come to mind in terms of how it can be used to the advantage of the human genome as we start to address diseases.
Well, if it is a disease that we know which gene is defective, and there are many, and we know what needs to be corrected, we could, not just in theory but in practice, go in and correct it.
So are we really talking about the elimination of disease down the line?
No, we're talking very specifically about only diseases caused by something genetic. So it's genetic diseases, and it's very specifically only a small category. It has to be caused by one gene. We're not going to be replacing 20 genes at the same time. And a lot of our genes interact. So for example, when you say cancer, can you cure cancer with it? Well, there may be types of cancer that are caused by one gene that could then be cured or dealt with.
So
but
the key
it's not
is
going to
isolating that gene? Is it
the key
isolating
is,
the gene?
but most diseases are not by a single gene. That's the key. So it will never cure all of the cancers. There might be a type that has one gene. Or blindness. I mentioned it earlier. There are some types of blindness caused by a single gene that we can fix. Other types are caused by multiple genes or things we don't even know about, and it won't work for those.
So we can't say that this will lead to the elimination of diseases.
No.
It will just help us in the treatment of some diseases.
Particularly because you're only talking about genetic diseases. I mean, you're not talking about all forms of pathology, infectious diseases. Somebody cautions you on the head, is that a disease? Yeah, I'm going to be pretty ill.
So we're not talking about the flu here.
No, not here.
Okay.
But I'd like to if you want to.
Okay. So just for the record, for the audience, the A, T, C, and G, we're talking about adenine, thymine, cytosine, and guanine.
Correct.
And so that's where the ATCG comes from. And these are nucleotides. Again, we're not doing a quiz at the end of the show, but just in case the audience wants to know, what are those letters in that alphabet soup? What do they stand for? What do they mean? So as we talk about genotyping the population, and there's already been some effort in that area, genotyping, and scientifically we can probably do this. The question is, what do we do with that information, and what are the ethics involved in the privacy of collecting that data? Because we know we have trouble with securing databases these days.
That's for sure. I mean, when we look at what's gone on with lots of different hospitals, healthcare systems, states, banks, and stores, yes, we understand that our information is not secure. And here we're talking beyond genotyping, we're talking sequencing. We're talking all of the ATs, Gs, and Cs. One of the goals had been to be able to make human sequencing something like a medical test. You walk in, you plunk down something under $1,000, you wait about 10 days, and you get your response, which in the case of rare and unusual diseases would be phenomenal. But it is, at the moment, a little bit frightening given that we have not yet dealt with how do we secure our data. And that's not a biological question.
Okay, so that's really a database management security issue.
It is.
More than it is a biological issue. Okay, so let's talk about...
Could I add one thing?
Sure.
Even though we still have that issue, a lot of people are currently being sequenced. Their data is out there. And that must be frightening because it has some other implications. Not only is it about you. So let's say they sequence you, they find out that you have the disease, you have the gene for Huntington's. Well, that's not only telling you information, but now your mother knows that her father died of this. There's only one way you got that gene. She may not have wanted to know, so it's going to create all kinds of issues within families because you're providing other people information perhaps they didn't want to know. And this has been going on ever since we've been doing any kind of gene analysis. You know, it goes back to the original BRCA gene, the breast cancer gene. And again, we have to make a really clear distinction here. There's two types of genetic genotyping gene testing. One type is to say, what gene do you have in your body? We know when it's broken in this way, it causes a disease. For example, BRCA, I mentioned it earlier. We've known for many, many years, women who have the BRCA1 mutation, the BRCA1 gene, they have something like a 90% chance of getting breast cancer. That's a really high risk to be running around with. Different from that, a lot of the genes we know, well, if it looks like this, then you have a 70% or a 50%, but maybe not. And the difference between the ones that you know you're pretty sure going to have a disease, or you absolutely do have the disease in the case of Huntington's, and it just expresses when you're older. Those are very different kinds of tests, and the public has to be ready for tests saying, yes, you have it. Like if someone biopsies you, looks at your cells and say, this is cancer. It's cancer. Very different from, well, you might develop cancer by being in the sun. Anyway, I just wanted to make the distinction between maybe sometime and you have it now. Those are very different states.
All right, well, let's talk about how this tool or these tools of genetic manipulation can be used pre-birth. Are we entering an era where parents can determine what their baby is going to, how the baby is going to come out in terms of eye color, in terms of resistance to disease or elimination of disease we can understand, but what about these other aspects of selectivity for traits that the parents may want? Everyone's going to want an Olympic athlete for a son or daughter.
Yeah, the pre-implantation, pre-genetic diagnosis has been around for many, many years. This is not new. This pretty much spun off after we had in vitro fertilization, because now we're not talking whole genome, we're talking single genes that you're looking for. So yes, you can, and have been able to for many years already, been able to take sperm and egg, put it together, look at the embryo that's beginning to grow, and ask the question, there's Tay-Sachs running in both of our families. If this embryo is implanted, if this baby is born, will this baby have Tay-Sachs? That question has been answerable for years, which means you can select the one without Tay-Sachs. Probably shouldn't be doing this, but you could also select the boy or the girl. This is all testable. Not legal, but the Tay-Sachs is legal. The other, not so much. This has been around for years. This is not new. That's what's so weird about some of this technology. The new piece is the CRISPR-Cas. That's only been used since about 2013. The ability to look at these things, been around for a long time.
But we're getting better and better at it. That's my point, is that where does it stop? Where do we draw that? You said the lines have already been drawn to a certain degree. Where do we draw the line as we know more and more and more about what various genes do?
I think we have to decide that as a society. I think that that comes down to political decisions. That comes down to what people are willing to do. I mean, I know that eventually you could go to, well, maybe we shouldn't be manipulating things in this way, but we've been manipulating genomes for forever. I mean, people talk about why dogs are such wonderful companions. They've been selected to be. Or how come our vegetables come out the way they come out? There's been selecting the best seed and breeding the best seed going on for forever. So this concept of manipulating genomes is not new. We're just taking it to a different level.
We're just more precise in terms of how deep we can get into the science, I guess.
I think it's more precise on exactly what we're moving and potentially bringing better cures.
Mm-hmm. Okay, all right. Well, let's talk about GMOs, genetically modified organizations, and food and agriculture. This has been really controversial in the last few years. A lot of people who favor organic foods and organic products are adamantly opposed to GMOs because they believe that these synthetically produced plants are not native. They cause problems. People have suggested that they cause digestive problems, allergies, that they're precursors to certain kinds of diseases like celiac disease or perhaps even type 2 diabetes. What about that?
I don't think there's tons of evidence of the GMOs doing anything bad with regards to allergies and diabetes. Because what a GMO is, let's first say, what is this? You know, not some magic little black box over there. A genetically modified organism means that you've moved a gene in. Like you can move a gene into strawberries. That makes them not freeze when the first frost comes through. That gene can be from anywhere. It happened to be from a fish, but it doesn't matter. It is one gene. Remember I talked about a gene as a word. A piece I didn't say is when I talked about these words and these letters, the stuff it's made of, DNA, the A's, T's, G's, and C's comprise DNA, they make DNA, it's a sugar. So unless someone's running around saying, well, it's bad for us, it's a sugar. So now, we're talking about we're adding a little piece of A's, T's, G's, and C's into another organism, genetically modifying it. Is it intrinsically bad? No. No, I think there have been some really interesting applications where it's been good. Golden rice, the idea that you can make beta-carotene to deliver vitamin A so that you'll have fewer children who are blind and much, much better immune response so they're not dying of disease. It's an amazing project. Flip side, when you look at what the majority of the GMOs have been in the United States, and I'm being very specific about the country, seems to me that a large, large proportion are to become pesticide and herbicide resistant. So what that means is you have a plant that you put a gene in. Putting the gene in didn't really change the plant. I doubt it changed much nutritionally, frankly. This has been looked at by a number of major organizations, including the Genetic Society of America. But what has changed is now I can dump tons of pesticides or herbicides on this plant, more than I used to, so you can't wash it off. So I'm much more afraid of the chemicals being dumped on than the modification itself. That would be where I would raise questions.
They've created hardier plants, as you just said, and much more able to resist pesticides as well as other things, and that's the danger, is so many chemicals now going into the process.
That is, and I think the other danger, which is somewhat unique to our country, most of the planet, I'm not going to say all of it, I don't know every single country's laws, most of the planet has labeling laws, so that it's actually labeled. And if you don't want to eat GMOs, you have a right to choose. In the United States, we don't have labeling laws. So you don't know what's in your products. And I can tell you that the GM soy, which is in a lot of our foods, is everywhere. So I think I would speak in favor of being able to at least, land of the free, we should be able to make our choices.
We just have a minute left, so I'm going to throw you a question and only give you 30 seconds to answer.
Great.
Lamarckian evolution and what's known as epigenetics. What is that?
Okay. Epigenetics, epi means above. So it's in addition to your A's, T's, G's, and C's that make the words that I talked about before. And what it is you can actually stick things onto your C's that make a gene either be turned on or not turned on as much. This can be influenced by the environment, and it can be passed supposedly from generation to generation, so it is heritable, sort of. The rules of heritability are not cleanly figured out yet, but what we do know is in the early mouse studies that mice could be two different colors just based on whether or not you had this modification. The other piece about the epigenetics, though, is that the DNA itself, the things that make the chromosome, the way it's stored in the cell, it's wrapped around these giant things that are like curlers, like your hair curler. You put a big wrap. You can modify the curlers so that it doesn't untwist the same. But again, we don't really know how many genes or which genes in which organism in a human would be affected by this or how long it would stay.
And with that, we have to bring the program to a close. I want to thank you for being with us today and explaining all of these difficult concepts.
Thank you for having me.
And thank you for joining us on this edition of Talking Points. Join us again next time when we explore another interesting topic. Until then, I'm Dave Kelly. Have a nice day.